In this presentation, we're going to explore the details of the light reactions in photosynthesis. And before we get into those details, I want to remind you that in many regards, photosynthesis is going to be the opposite of what you studied in cellular respiration. For example, if we look at free energy during the entire process, photosynthesis is going to be a process that requires an energy input. Therefore, photosynthesis is going to be a set of overall endergonic reactions, whereas cellular respiration overall ends up releasing free energy, and that is going to be an exergonic kind of a reaction. Also, photosynthesis is going to be the opposite of cellular respiration in terms of redox reactions. Like cellular respiration, photosynthesis involves a series of transfers of electrons. When a substance gains electrons and hydrogen ions, is being reduced. When a substance loses electrons and hydrogen ions, is being oxidized. And what happens exactly with photosynthesis is that carbon dioxide is going to be reduced. Carbon dioxide is going to be gaining electrons and hydrogen ions, and it's going to become glucose. So you can say that carbon dioxide is reduced to form glucose. What is the source of electrons that are going to be given to CO2? Well, it's going to be water. Hydrogen ions and electrons from hydrogen that used to be in water will be given to CO2, and as water loses those hydrogen ions and electrons, water will be oxidized and is going to form oxygen gas, which is that good oxygen gas for us because we breathe it and because we need it for the purpose of cellular respiration. So remember, from a redox point of view, in photosynthesis, carbon dioxide is reduced to form glucose and water is oxidized to form oxygen. Now that I said those important details, let's take a look at the light reactions. Remember that the light reactions is going to be one of the two main stages we see in photosynthesis. The light reactions take place in a very special type of membrane located inside of the chloroplast. Thylakoid membranes are going to be like folded membranes, and they fold into what looks like stacks of pockets. And so those stacks of pockets, they look green when you see these with the light compound microscope. And that is because this is where the photosynthetic pigments needed for harvesting light, for capturing light, are going to be housed. So in the light reactions, light is absorbed. Electrons will be taken from water. Water is going to be oxidized and becomes just oxygen. The electrons from water are going to be loaded into an electron shuttle NADPH. The name is familiar to what you saw before in cellular respiration. NADPH stands for nicotinamide adenine dinucleotide, and in this case, the P means phosphate. So NADPH is going to be carrying pairs of electrons to the second stage of photosynthesis, which is the Calvin cycle. At the same time, during the light reactions, there's going to be energy that is going to be captured and is going to be stored temporarily in ATP. ATP will provide energy for the reactions of the Calvin cycle. So notice that there are two main contributions of the light reactions to the overall process of photosynthesis. The light reactions contribute ATP energy, and they contribute high energy electrons transported by NADPH. And remember also that in the light reactions, water was oxidized. It gave off electrons and hydrogen ions, and it became oxygen. This is important because the light reactions is what is providing the energy and the electrons necessary to run the Calvin cycle. The Calvin cycle, I'm just going to mention it right now in passing, is going to be taking place in the stroma. The stroma is that thick fluid, like jello-like fluid inside of the chloroplast. And the main function of the Calvin cycle is going to be reducing CO2. CO2 takes electrons and hydrogen ions and it's going to become sugar. It's going to eventually lead to the formation of glucose molecules. But let's go back to the light reactions. The light reactions are going to need, well, of course, light. What specific light are we talking about here? We're talking about all of the wavelengths that fall within the visible light spectrum. And so that is going to be from violet all the way to red. So colors like blue, green, yellow, orange, all of those colors are going to be absorbed during the light reactions. Notice also that visible light is part of the family of energy we call electromagnetic radiation. And there are other examples of electromagnetic radiation in this illustration, but we're only interested in what we call the visible light spectrum. How do we capture light? Plants are going to be capturing light with the help of photosynthetic pigments. That is the mission of a photosynthetic pigment. Obtain light, capture it, and there are different types of photosynthetic pigments. For example, chlorophyll A and chlorophyll B are easy to recognize because they look green to our eyes. But why do they look green? If you look at this illustration, this is the chloroplast. Again, this will be the thylakoid membranes, like stacks of pockets. There'll be a stroma medium inside. And so when light shines through the two membranes of the chloroplast, will hit the photosynthetic pigments. Notice that in this case, green light is being reflected. That is exactly what happens when light shines on chlorophyll A and chlorophyll B. These two pigments do not absorb green light. It's reflected, and that's why we can see it. Chlorophyll A and B can absorb red, orange, yellow, blue, violet light. And so what happens during the day if green light is being reflected? It's like this type of light will not be usable by the plant if the plant only has chlorophyll A and B, and that would be like a waste of energy. Plants don't want to do that. When the sun is shining, plants want to absorb every possible wavelength within the visible light spectrum. And so what do plants have to capture at least some of that green light? And the answer is real clever. Have other pigments that look a different color than green. So for example, carotenoids. Carotenoids can range in color from orange all the way to red. Carotenoids, because they look orange or red, means that they're reflecting orange light or reflecting red light. And that means that they are absorbing green light. So in combination with chlorophylls A and B and carotenoids, plants can do the job of absorbing all of the visible light wavelengths of color. And some plants also have xanthophylls. Xanthophylls look yellow to us. And remember, if they look yellow, means that yellow light is being reflected, but a xanthophyll can absorb green and red and blue and violet. Photosynthetic pigments don't work alone. They are going to be part of a larger complex we call a photosystem. The photosystem includes photosynthetic pigments and proteins, and the main mission of a photosystem is going to be harvesting light, not only absorbing it, but retaining it, keeping it, and transforming it into another form of energy that will be usable for the ultimate purpose of making glucose in photosynthesis. So what are the exact components of a photosystem? First, we have an antenna assembly. This is usually like a cluster. As you see in this illustration, many photosynthetic pigments are going to be clustered together very close in proximity, about 200 to 300 of these pigments, mostly chlorophylls, by the way, but mixed in there we have some carotenoids and some xanthophylls as well. There's going to be a pair of modified chlorophyll A molecules known as the reaction center. And in a photosystem, you'll also see a protein complex known as the primary electron acceptor. We are about to look at exactly how all of these parts contribute to the harvesting, to the keeping, trapping of sunlight. I do want to mention also that there are two types of photosystems in most plants. And you can see in this illustration the two kinds of photosystems. One is called Photosystem II, Photosystem I. They all share the three components we said before. Notice that when we look at the light reactions, Photosystem II participates first. And some of you will be like, what's the deal here? Did someone mess up with the naming? Why did they call this photosystem Photosystem II if it's actually the first one to participate? Don't stress too much about this. Simply the names Photosystem I and Photosystem II were given based in the order in which scientists discovered the presence of these light harvesting units in a plant. So you can say Photosystem II was discovered second, later, time after Photosystem I had been discovered. So don't get too caught up in that particular detail. But it is important that you know, if I'm talking about Photosystem II, that this is one where we begin to follow the light reactions. So when light shines, and if you can look at this illustration again on the left-hand side, here we have light traveling through space in the form of photons, which are discrete packets of light energy. Photons will go through the leaf epidermis layer of cells and go through the mesophyll cells and eventually penetrate through the membranes of the chloroplast, reaching the thylakoid membrane. When light hits the photosynthetic pigment like chlorophyll, electrons are going to get excited. And the way electrons get excited is that an electron goes from a lower energy level to a higher energy level. Now, the detail about excited electrons is that they don't stay forever in this excited state. After it's been in a higher energy level only briefly, the electron returns back to a lower energy level. And when it does so, it releases free energy, and that free energy is going to be transferred to the next photosynthetic photosynthetic pigment. This pigment is also absorbing light, and electrons are moving to a higher energy level, returning to a lower energy level. That releases more energy. And so this energy of resonance, which is what we call it in chemistry, is being transferred from pigment to pigment to pigment until eventually all that energy is captured by the reaction center. Here, the reaction center will have one of its electrons also get energized, excited to a higher energy level, but it does more than that. This one electron is actually going to jump to another molecule, which is going to be this protein complex we know as the primary electron acceptor. And from here, this electron is going to be moving down an electron transport chain very similar to the electron transport chain we studied in cellular respiration. The purpose of this electron transport chain is going to be allow electrons to move from a higher energy state to a lower energy state as it moves through electron carriers and special proteins called cytochromes to another electron carrier, and then it's going to reach eventually photosystem I. Remember that as energy is being given off by electrons moving down an electron transport chain, that free energy can be used for creating a hydrogen ion gradient. And this hydrogen ion gradient will be used for the making of ATP through that process called chemiosmosis, which we're going to review again in just a moment. So what happens when the electron was excited comes here to photosystem I? Well, the sun is still shining. Energy from the sun is still exciting electrons. This energy is being transferred across these molecules in the photosystem. And eventually all that energy will excite the electron that came from photosystem 2. Now that electron is here, and that electron is going to be energized and transferred to the primary electron acceptor of photosystem 1. And here, this electron will go into another an electron carrier, and it's going to be loaded into the coenzyme NADP plus a hydrogen ion and one electron. It's going to become NADPH. So when two electrons have arrived here, NADPH can carry those electrons over to the Calvin cycle. That's how the light reactions work. Light energizes electrons. Electrons move down an electron transport chain. That free energy they are giving off is used for the making of ATP. And eventually, a pair of high energy electrons will be loaded onto NADP+, and it's going to turn into NADPH, carrying a pair of electrons to the Calvin cycle. Those are the stages of the main steps in the light reactions. Some other important details that we need to pay attention to. Let's take a look at this illustration. This is like a thylakoid membrane, one of those pockets I mentioned previously. Here, for the sake of simplicity, we're only showing two photosystems, but there's going to be many of these photosystems on any one of those pockets of the thylakoid membranes. And so here we have photosystem 2, which is where electrons are going to be energized. Now, one thing that would happen is that as this electron leaves photosystem 2, now photosystem 2 is going to be short one electron, and it cannot stay in that unstable type of a status. So almost immediately, water will be split to give back the electron photosystem 2 sent along this electron transport chain. When another electron departs from photosystem 2, another one of the electrons from water is going to be given to photosystem 2 so that it can replenish the electron previously lost, which was sent down this electron transport chain. And so this photosystem 2, in biology, we also call it the water splitting photosystem, because this is the place where water will be split to provide electrons that are being sent ultimately down the electron transport chain and eventually transported through an NADPH. What happens to water when it loses the two electrons? There are going to be two hydrogen ions. Two hydrogen ions, one will be staying here inside of the thylakoid space, and eventually some of them will move out, and one of the hydrogen ions will be picked up by NADP+ to become NADPH. So water is our ultimate source of electrons in the light reactions. Another important point, NADPH is going to be delivering electrons for the redox reactions of the Calvin cycle. And finally, let's take one more look at chemiosmosis. It's similar to what we saw before in cellular respiration, but there are a few differences, because here chemiosmosis is taking place in the thylakoid membrane. Before in cell respiration, chemiosmosis happened in the mitochondrion as hydrogen ions reached a high concentration in the space between the inner mitochondrial membrane and the outer mitochondrial membrane. That was cell respiration. That was in the mitochondrion. Here, the high concentration of hydrogen ions is being built inside of this thylakoid pocket. We can call this the thylakoid space. When a high concentration of hydrogen ions is built, these hydrogen ions have to depart. They have to move away from this thylakoid space, and the only way they can escape is going to be through a port provided by this protein we've seen before, ATP synthase. ATP synthase allows for hydrogen ions to move through, and as those hydrogen ions are moving, we know that ATP synthase turns like a turbine, and that somehow, we don't know exactly how the mechanism works out, but we know that that is what allows for adenosine diphosphate to be joined by another phosphate so we can make adenosine triphosphate, the energy molecule that supplies power for all of the work cells need to do. And in this case, ATP will be powering the next stage of photosynthesis, which will be the Calvin cycle. So that's all for right now in terms of the light reactions. Oh, I should mention also the oxygen that resulted from the splitting of water, that oxygen is going to leave the chloroplast, is going to leave the plant cell, and it has to go out of the leaf because oxygen during the process of photosynthesis can reach high levels and it can be toxic to the plant. So I'll make another video to explain to you the details of the Calvin cycle.